Welcome back to the deep dive. If you've ever tried to follow the science of brain repair after something major, like a stroke, well, you know how tangled it can get, fast. For decades, when neurologists looked at the brain after an injury, they saw this thick, fibrous wall building up around the damaged spot, the glial scar. And the thinking was, this scar is the enemy. It's this physical roadblock, right, put up by these support cells, astrocytes, stopping nerves from regrowing, just getting in the way.
That was definitely the prevailing idea, very mechanical view. But the evidence now really paints a different picture. It's shifted quite a bit.
Oh, how so?
Well, these astrocytes right at the border, they're not just passive bricklayers. They're actually sophisticated regulators. They're deciding, molecule by molecule if that area heals well or if it just dives deeper into damaging inflammation.
Okay, so the question isn't how do we bulldoze the scar anymore? It's more like how do we tweak it, adjust the signals.
Precisely. And that's our mission for this deep dive. We're looking at research that use some incredibly high-resolution tools, single-cell analysis, things like that, to find the exact molecular switch controlling that inflammation.
And there's a specific culprit.
There is a protein called BST2. It's expressed by just a small, very specific group of astrocytes right at the lesion edge, and it seems to act like a gatekeeper, unfortunately opening the floodgates for destructive inflammation really early on after the stroke.
A gatekeeper protein. Okay, BST2.
Yeah, and finding this gives us a concrete target. So today we'll trace that chain of command. How BST2 kicks off signaling, specifically using the C3-C3-IR pathway, which is, well, notorious in inflammation circles to recruit microglia that actually cause more harm.
And the payoff.
The payoff is that the research suggests blocking just this one protein, BST2, could be a really promising way to help the brain repair itself, especially in those critical early days.
Okay, let's get into it. Before BST2, the protein, we need the cell. You mentioned astrocytes, but they're not all identical, are they? How did researchers even zoom in on the right type of astrocyte?
Right. Astrocyte diversity is huge. They needed something like a molecular fingerprinting kit. That's where single-cell RNA sequencing, or scRNA-seq, comes into play. It lets you look at the genes active in thousands of individual cells.
So you take the injured brain tissue. When exactly?
At critical time points, day 1, day 3, day 5, day 7 after the stroke. This is crucial because it's before that glial scar fully forms, which usually takes about 2 weeks. You want to catch the action as it's unfolding.
And this key RNA-seq, it tells you what genes are switched on or off in each cell.
Exactly. It's like giving each astrocyte its own detailed profile based on its gene activity. This allowed the researchers to sort all these reactive astrocytes into 6 distinct groups or subclusters. They labeled them ASER to AS5. 6
different personalities, essentially.
Kind of, yeah. And this detail is vital because, most of these astrocyte types are probably doing helpful things, necessary repair work. The challenge was finding the needle in the haystack, the one subcluster driving the damaging inflammation.
So out of the 6, which one emerged as the main suspect, and how did they pinpoint it?
The spotlight fell on the AS2 subcluster. Its numbers were significantly up after the injury, consistently. Then they used computational methods, like cell chat.
Cell chat? What's that?
It's a tool that predicts how cells might be communicating by looking at which ligands signaling molecules one cell type makes and which receptors another cell type has to receive those signals.
Like mapping out the potential phone calls between cells.
Exactly. And cell chat analysis showed that AS2 astrocytes were predicted to be talking the most and the loudest, specifically to microglia. Microglia are the brain's main immune cells, so AS2 was doing the most shouting at the immune system.
Okay, AS2 is the talkative one. What specific message, what molecular signal were these AS2 cells sending that linked them to inflammation?
Well, their gene profile showed they were packed with genes from something called the M1 module. Now, M1 genes are often associated with driving a pro-inflammatory, sometimes damaging immune response. Think of it as the attack mode gene set.
So they're genetically primed for aggression?
In a way, yes. And when the researchers looked deeper into the protein interactions within this M1 module in the AS2 cells, one protein stood out right at the center of the network, like a key hub. That was BST2 bone marrow stromal cell antigen 2.
BST2. What's its usual job? Why is it popping up here?
The ST2 is interesting. It's known primarily as an interferon-inducible protein. That means its production gets ramped way up when cells sense interferons, particularly type I interferons, which are strong immune alarm signals.
So the presence of BST2 tells us these AS2 astrocytes are actively responding to immune alarms.
Precisely. It led the researchers to classify these AS2 cells as a specific subtype, interferon-responsive reactive astrocytes, or IRRAs. They're not just bystanders caught in the crossfire. They are direct participants, amplifying the immune response.
An IRRA. Okay, that gives the cell a clearer identity. But in the brain, location is everything. Where exactly were these BST2 positive IRAs physically located?
That's where spatial transcriptomics came in handy. It's a technique that lets you map gene activity back onto the physical structure of the tissue, combining that with immunofluorescence staining.
Which lights up specific proteins in tissue slices.
Right. They confirmed that these BST2 positive astrocytes were highly concentrated. They only made up about 10% of all the reactive astrocytes, so a minority, but they were packed incredibly tightly right at the immediate border of the stroke lesion,
How tight?
within a zone just 0 to 400 micrometers from the edge of the damage.
Ah,
Well, it's really narrow, thinner than maybe 4 human hairs stacked together.
wow, so it's not spread out, it's targeted right at the interface.
Exactly, a really sharp boundary. And this intense concentration started to appear around day 5 after the stroke, which fits perfectly with when the glial scar itself starts to mature and organize.
And does this pattern hold up beyond mice?
That's a key point. They checked human brain samples from stroke patients and also from patients with traumatic brain injury, TBI, and they found the same thing. BST2 highly enriched in astrocytes right at that lesion border. This suggests it's a conserved mechanism, not just a mouse phenomenon.
Okay, let's pause for a quick recap for everyone listening. We've zeroed in on the troublemaker, a specific type of astrocyte called an IRA belonging to the AS2 subcluster. We found it using its genetic fingerprint high in M1 genes, especially BST2, and we located it packed tightly in that tiny 0-400 micrometer zone right at the stroke border, starting around day 5. Its calling card is the protein BST2. Got it.
Perfect summary. So now we know who is potentially causing the problem and where they are. The next question is how. How does BST2 on these astrocytes lead to damage?
Right, what's the mechanism? You mentioned microglial earlier, the brain's immune cells. How they fit in?
Well, the researchers observed that microglia, which they identified using the marker IBA1, weren't just randomly scattered. They were clustered, aggregated very densely, right next to these BST2 positive astrocytes.
So they're drawn to them.
It certainly looks that way. The highest density of microglia was found in the inner part of that border zone, the 0 to 200 micrometer region, even closer than the astrocytes themselves. And the number of microglia there correlated directly with the number of BST2 plus astrocytes over time. It was like the microglia were following the BST2 signal.
Like heat-seeking missiles locking onto the BST2 beacon. What is that beacon signal? How are the astrocytes calling them in?
That led them back to analyzing the communication pathways, digging deeper with tools like cell phone DB. And the strongest predicted link, the loudest molecular shout between the AS2 astrocytes and the microglia, involved the complement system. Specifically, the C3-C3R pair.
C3. Component 3, that's a major player in inflammation.
Yeah.
Right. Often seen as bad news in the brain.
Very often, yes. It's a powerful inflammatory mediator. And the analysis showed the BST2 positive astrocytes were the primary source. They were co-localized with high levels of C3 expression, that they were essentially pumping out C3.
Okay, so the astrocyte makes C3. What about the microglia?
The microglia accumulating at the border were found to have high levels of the receptor for C3's active fragment, C3A. That receptor is called C3AR. So the astrocytes are sending the C3 signal, and the microglia right there have the antenna, C3AR, to receive it loud and clear.
And these C3R positive microglia, are they just hanging out or are they activated?
They're highly activated, showing markers associated with a damaging pro-inflammatory state. They're not the helpful cleanup crew microglia. They seem to be contributing to the problem.
Okay, but hang on. BST2 is a protein, you said, often found on the cell surface. How does a protein sitting on the outside of the astrocyte tell the machinery inside the cell to start mass-producing and spitting out a completely different protein like C3? There must be steps in between.
Absolutely. That's the core of the mechanism they uncovered. It's not just that BST2 is there. It's actively doing something inside the cell. It involves a couple of key intracellular signaling molecules, protein kinase C-beta-2 or PKCβII, and the infamous NFU pathway.
Okay, PKC and NFUV, these sound like important internal switches. Walk us through it.
So the study found evidence that BST2 on the astrocyte surface acts as a regulator. It physically interacts with, it binds to, PKCO inside the cell. And this binding promotes the phosphorylation of PKCA2.
Phosphorylation. That usually means activating something, right? Like flipping a switch.
Exactly. Think of PKC2 as an enzyme that's normally inactive. BST2 binding helps add a phosphate group to it, which switches PKCU on.
Okay, so BST2 activates PKCU. What happens next? Where does NFD come in?
Once PKCU is switched on, its downstream target is the NFB pathway. NFB is like a master control system for inflammation and immunity genes inside many cells. Activated PKCU triggers a cascade that ultimately activates NFC.
And how do we know NFB is actually activated?
They looked for the telltale signs. One key sign is seeing the main component of NFB, a protein called P65, move into the cell's nucleus. The nucleus is the control center where genes are read. So P65 moving there means it's ready to turn genes on. They also saw phosphorylation of other proteins upstream in the pathway, like ICO and IBA, which are necessary steps to release P65.
Okay, so P65 is in the nucleus, ready to issue commands. Why does that specifically lead to C3 production?
Because the gene for C3 is a known target of NFG. When NFG gets activated in astrocytes, one of the main genes it switches on is the C3 gene. So the researchers saw a huge increase in C3 expression, and this increase was dependent on that NFG activation.
Wow. Okay, so the whole chain is BST2 on the surface activates PKC2 inside wing, which activates NFP, which goes to the nucleus and tells the cell, make lots of C3.
You got it. That's the sequence. BST2 isn't just a marker, it's the trigger for this whole internal cascade leading to C3 secretion.
Let's try that analogy again for everyone listening. Imagine that BST2 protein is like a megaphone bolted onto the surface of these specific IRA astrocytes at the border. It's constantly amplifying an internal danger signal.
Good analogy.
And this forces the astrocyte to shout, C3, C3. This C3 signal travels out and hits the C3R antennas on those nearby microglia. And receiving that signal transforms the microglia. They shift from potentially helpful cells into aggressive neurotoxic responders. Researchers sometimes call this a dam-like phenotype disease-associated microglia. Basically, they become part of the problem.
A very accurate picture, and the consequence of this toxic communication is exactly what you'd fear. The study documented that this whole BST2-C3 axis led to worse outcomes early on. More neuronal death, fewer healthy neurons surviving near the border, and more neurons undergoing apoptosis or programmed cell death.
And that translates to function.
Directly. It resulted in worse motor function deficits in the animals during that critical early recovery period.
Okay, so if BST2 is the ignition key, turning on this whole destructive engine, then taking the key away should stop it, right? Did blocking BST2 work?
It did. They tested this first with the genetics, creating what's called a conditional knockout mouse. They specifically deleted the gene for BST2 only in astrocytes. These were the BCTCKO mice.
And what happened in these mice after a stroke?
The results were pretty dramatic. In the BST2-CKO mice, the astrocytes produced much, much less C3. That critical signal was turned way down.
And the effect on the microglia in the brain?
With less C3 signal, the microglia didn't aggregate as densely at the border. The ones that were there looked different, less activated, more branched, more like resting microglia. And most importantly, functionally, these mice did significantly better. They had smaller infarct volumes, less brain damage, and showed much better recovery on motor tests like walking on a grid or staying on a rotating rod, especially in those first 2 weeks.
That's strong evidence BST2 is the driver. Did they double-check that C3 was really the crucial link after BST2?
Yes, they did a crucial control experiment. First, they showed that overexpressing BST2, forcing astrocytes to make even more of it, made the stroke outcome worse, as expected. But then, they did something clever. They took the BCPKO mice, the ones that were doing better because they lacked BST2, and in those mice, they artificially forced the astrocytes to produce C3 again, bypassing the need for BST2. And guess what?
The benefit disappeared.
Completely. As soon as C3 production was restored, even without BST2, the mice lost all the protective effects. They were back to having worse outcomes. This really nails C3 as the essential downstream effector molecule that BST2 works through to cause harm.
Okay, genetic deletion is great proof, but not exactly a therapy for humans.
Yeah.
Did they try a more drug-like approach? You mentioned BST2 is also called CD317.
That's right. CD317 is another name for BST2. So they developed a monoclonal antibody designed to specifically bind to and block CD317-BST2. They administered this antibody to normal mice right after inducing a stroke.
Like giving a drug. How did they give it?
They injected it intraperitoneally into the body cavity at a specific dose for the first 5 days after the stroke, targeting that acute inflammatory window. The idea was to see if this antibody could replicate the benefits they saw in the genetically modified mice.
And did it work? Did the antibody block the whole destructive pathway?
It seemed to work remarkably well. On the molecular level, antibody treatment reduced the signs of PKC2 and NF-κ activation inside the astrocytes, and consequently, it lowered C3 expression, just like the genetic knockout.
And cellularly,
Yeah.
microglia, neurons.
Cellularly, yes. They saw fewer activated microglia clustering at the border, less staining for markers like MAC2 and CD68, which indicate activation. And importantly, there was less neuronal loss in the antibody-treated mice.
Fantastic. And the bottom line, did it help the mice recover better?
It did. Functionally, the mice treated with the anti-CD317 antibody performed significantly better on those motor tests, grid walking, rotarod, pole test indicating improved neurological recovery in the early phase. It essentially phenocopied the genetic knockout.
That sounds incredibly promising as a potential therapy.
Yeah.
But you hinted earlier there was a catch, something about timing.
Yes, and this is a really critical point for any potential translation. The benefits of blocking BST2, whether genetically or with the antibody, were only seen during the early phase after the stroke,
How early?
up to about day 14. When they looked later, say at day 28, the differences between the treated and untreated groups had largely vanished. The benefits were confined to that acute-subacute window.
Why would that be? Why would blocking BST2 help early but not late? Does C3 maybe do something different later on?
That's the strong implication. The BST2-C3 axis seems to be specifically driving harmful inflammation early, but the complement system, including C3, is complex. Later on, during the chronic repair and remodeling phase, C3 and its receptor C3AR might actually play different roles, potentially even beneficial ones, maybe related to clearing debris or synaptic plasticity.
So it's not just C3 bad, it's C3 bad right now in this early phase.
Exactly. It underscores the idea that you can't just shut down the complement system entirely or indefinitely. That could interfere with long-term healing. Any therapy targeting this pathway, like the anti-BST2 antibody, would likely need to be phase-specific, delivered only during that initial window where BST2-driven C3 production is clearly detrimental.
Okay, so let's wrap this up. This deep dive has really changed the perspective on that old glial scar. It's not just a wall, it's an active communication hub.
Definitely. And this research pinpoints BST2 on those specific IRRA astrocytes as a key molecular switch. It flips those astrocytes towards promoting neurotoxic inflammation by cranking up C3 production, which then activates microglia via the C3RR receptor.
And the therapeutic angle is quite exciting. Instead of trying broad anti-inflammatory drugs or risky systemic complement inhibitors, this anti-CD317 antibody offers a potentially much more precise way to intervene.
Right. It allows for targeting the specific cell type, IRRAs, driving the problem right at the location where it matters, the lesion border, and potentially only during the time window, early acute phase when it's harmful. That specificity is really appealing.
We follow the whole chain reaction. It starts probably with type I interferon signals boosting BST2,
Which then activates the internal PKCOFU and NF-κ pathway,
causing the astrocyte to pump out C3.
which activates nearby microglia via C3R, leading to neurotoxicity and functional deficits.
So thinking ahead, if BST2 expression is kicked off by type I interferons, IFNI, that brings up a big question for you, our listeners, to ponder.
Where does that initial IFNI signal actually come from right after the stroke? Is it generated locally by dying brain cells? Or does it come from the systemic immune system maybe leaking in through a damaged blood-brain barrier?
Because if we could figure that out, maybe we could target that upstream signal even earlier. Could we prevent the BST2 gatekeepers from even being installed on those astrocytes in the first place, stopping this whole destructive cascade before it even begins? Something to explore.